This meeting is being recorded.
Good morning, everyone. My name is K. Sumuk Ediar. We are here to present our project topic Early Detection of Yellow Leaf Disease in Areca Tree under the guidelines of Professor Bobya ma'am. My teammates are Kiran V, Kiran Prasad, and Manjunath K.S. Introduction. Agriculture played a key role in development of Indian economic system. If there is decrease in farming, then the total economy of country will get affected. In today's era, farmers are facing many problems regarding fungal infection or bacterial diseases. Yeah, wild is major found in Sringeri, Koppa, and Surya in Karnataka. Leaves starting turning into yellow color from the tip towards the midrib. The main duration of yellow leaf diseases is from August September to April May. The major causes of the yellow leaf diseases is stunted growth and yellowing of root damage. Probably root were infected with pests, fungi, bacteria. After the severe infection, roots stop growing and start decaying badly. As a result, lateral roots start growing. When the root tips were damaged, there were clear indication of lateral roots formation. This could be the reason when roots were damaged. Yellowing started, and when lateral roots were developed, plants started taking nutrition, thus the plants is recovered. Further concept will be explained by Kiran Prasad.
Thank you, Sumuk. Let's see about the existing system. There are some approaches to detect yellow leaf disease by using the image processing technique and molecular detection of phytoplasma, which are associated with yellow leaf disease. And by using image processing technique also, we used to detect the disease after the yellow spot starts and the appear of the top lip tip. In molecular detection, all the required samples of the areca are taken to a lab and tested to check whether the phytoplasma is involved. If it's involved, they can cause the tree if the disease is infected or not by the yellow leaf. The proposed system will explain by our teammate.
Thank you, Kiran. Our aim of the project is to predict yellow leaf disease in Areca nut tree in the early stage itself and provide the solution to recover from disease. And we plan to build our project using machine learning by using some classification algorithm to classify the sample based on the model we create. We are thinking of using ID3 algorithm to build the decision tree that classifies the sample and predict the disease and providing an user-friendly interface so that the person with lesser experience in software should also be able to use it easily. We will also recommend which pesticide pesticide to use the affected tree. And next concept will explain by Q.
Thank you, Manjunath. Now I'll going to explain the objectives of our project. One of the main objective of our project is to predict the yellow leaf disease at the earliest and help the farmers to prevent the further spread. And we are trying to collect the accurate data set that contains all the attributes which causes the yellow leaf disease. And we use using appropriate algorithm to make a decision over the data set collected. And we'll try to find the exact number of data set required for classification using the algorithm. Next slide. Now we'll go on to literature survey. Paper 1. The title of our paper is Areca nut diseases and their management. Authors are Manimekalai Ramaswamy, Samita Nair, VP Soumya, and George V. Thomas. The journal is Indian Journal of Areca nut Spices and Medicinal Plants. In this paper, authors have summarized an outline of Areca nut disease and disorders and their management. They have reported about the following diseases. They are fruit rot, bud rot, and yellow leaf disease, inflorescence daybreak, anaberoga, leaf blight, collar rot, bacterial leaf steps. Our concern is YLD, that is yellow leaf disease, and they have mentioned about the yellow leaf disease symptoms. They are yellowing starts from the tip of the leaflet and It spreads to the outer leaves and sometimes it is seen in the middle or in the advanced stages. Yellowing spreads to all the leaves completely and dry and fall off. And the kernel of the nuts of affected palms will become soft and show blackish discoloration and assumes a spongy texture. And they have mentioned that yellow leaf disease is spread by or transmitted by a plant hopper called Pro- Protusta monesta. And for management of the disease, they have said that removal of the severely affected palm with a poor yield, completely removing it. And for nutritional management, they have mentioned that NPK at 100 is to 40 is to 140 per gram per palm per year. That is nitrogen, potassium, and nitrogen, phosphorus, and potassium. And organic manure at 12 kg per palm per year, and additional dose of super phosphate 1 kg per palm, along with the provision of summer irrigation. And they have mentioned of proper drainage facilities that should be provided to avoid water stagnation. Next paper. In this paper, the title of this paper is Yellow Leaf Disease of Areca Nut, Present Status in Karnataka. Authors are Jose C.T. D. Jagannathan, Journal ResearchGate. In this paper, they have mentioned about the yellow leaf disease spread and intensity in the all over in Karnataka. The mainly yellow leaf disease is spread in wait one minute. Sir, in first paper we got to know about the symptoms of this yellow leaf disease. We understood the symptoms and spread of this disease. Sir, we can improve by asking some specialist in that area, like agricultural specialist will be there in agricultural office, and asking farmers who are well known about the disease. Yes, sir. The early symptoms are from the early symptoms we can make a data set out of it, sir, to predict. Okay, sir. That's the title of a second paper is Yellow Leaf Disease of Areca Nut and Present Status in Karnataka. In this paper, authors have mentioned about the intensity of the yellow leaf disease in the state of Karnataka. They have mentioned the disease was first observed after heavy flood in 1914 from central Kerala. The Department of Horticulture Karnataka reported in 2014 that yellow leaf disease present in 4 taluks of Chikmagalur districts, that is Sringeri, Koppa, Narsimrajapura, and Mudigere, and Madikeri taluk of Kodagu district. The field survey conducted during November to December in 2014 revealed that yellow leaf disease is present in 65% of the Areca gardens in the disease affected area. In these taluks, and also it is estimated that it is present in 32% of the total Areca nut farm in the affected area. And they have found that highest disease incidence, that is 73%, as well as disease intensity 50% was in Madikeri Taluk, and the lowest disease incidence was observed in Sulia Taluk, that is 53%, while the lowest disease intensity was in Mudigere Taluk, that is 16%. From this paper, we can use the taluks as for our data set. For if a farmer lives in Madikeri, then there will be a highest intensity of the yellow leaf disease. Next paper will be explained by Sumuk.
Thank you, Kiran. Paper 3. Title of the paper is phylogenetic analysis identifies a candidate's phytoplasm aureus related strain associated with yellow leaf diseases of Areca palm in India. The author are Me- Meninakalai Ramaswamy, Smita Nair, VP Soumya, and George V. Thomas. Journal International Journal of Systematic and Evolutionary Microbiology. In this paper, researchers from Molecular Biology Laboratory, Central Plantation Crops Research Institution, Kasargodu, Kerala, have used to plant materials like spindle leaf samples from 15 mildly symptomatic palms, 5 each at the early stage, middle stage, and advanced stage of infection were collected from Sule district, Karnataka. A healthy sample was collected from Kasargodu was used as a negative control and did the phylogenetic analysis and the characterized the 16S RNA and section A gene sequence and assigned the organism to India YLD cytoplasm. 16SR group and subgroup classify of the Indian YLD cytoplasm was done using the computer simulated RFLP approach. RFLP means restriction fragment length polymorphism is a technique that exploits variation in homogeneous DNA sequence. Based on this phylogenetic analysis of 16 RNA and section A gene sequence, identify the India's YLD cytoplasm as a CA dot cytoplasm or Z related strain. And next paper, data of 16S RNA gene amplification amplicon based metagenomic signature of Areca nut rhizosphere soil in yellow leaf diseases endemic region in India. Authors are S. Poliraj, Ravi Bhatt, M.K. Rajesh, S.V. Ramesh, U.K. Priya, and R. Thava Prakash Pandan, Vinayaka Hegde, and P. Chaudappa. Journal are elsewhere. In this paper, the rhizosphere samples were collected from YLD endemic, apparently healthy rhizosphere soil and YLD endemic non-rhizosphere soil from Sulya Taluku, Karnataka. Palms is about 30 to 40 years of South Canara local cultivator, predominantly growth in that area, were selected. Samples were collected from the active root zone, 30 to 45 centimeter away, 30 to 45 centimeter away from the trunk core, and at 5 to 40 centimeter depth, where the root system was denser from the surface. The samples were processed and stored in the refrigerated condition for further downstream analysis. Next to DNA extraction, the sample roots with rhizosphere soil particles attached were placed in the sterile tubes containing 9 ml of psychological solution. The tube were vortexed for 5 minutes to detach the soil particles and then centrifuged at 400 rpm for 5 minutes. The supernatant was discarded and the remaining soil fraction was used for DNA extraction using the metagenous soil DNA extraction kit. Next, result analysis. The data quality was data quality of the raw reads was checked. The read was trimmed 20 bp and remove the degeneration primers. The trimmed reads were pre reads were processed and remove adapter sequence and low quality base using trim galore. Of the contiges were screened for errors. Only those between 300 bp to 500 bp were retained. Any contiguous ambiguous base calls was rejected. The high quality contiguous were checked for identical sequence and the duplicates were merged. After this process, we can after this process Protobacteria, bacteria, Actinobacteria, and other organisms are found in the rhizosphere of the arecanut palms, which is infected in yellow leaf diseases. Next paper will be explained by Manjunath K.S.
Thank you, Sumak. On paper 5, the title of the paper is The Prevalence of Yellow Leaf Disease and its Associated Areca Palm Vela Virus in Betel Palm Plantation in Hainan, China. Authors are Ang Jing Ang and Rubia Zhao, and journalist, agronomy. This study is based on the PlanetScope images to carry out the remote sensing monitoring of Areca yellow leaf disease for large-scale regional, and results of domestic of RF model exhibits the highest overall recognition accuracy of Areca yellow leaf disease 88.24% and 2.95% and 20.5 higher than the BPNN and ADA. Areca yellow leaf disease is the major attack of the planting and the production of areca nut. The study area is located in the Bidia town, meaning country and province of China. Considering the pixel size of the remote sensing images, areca nut planet securely of the disease survived. Track IS and orbital invention sensor, type width and spatial revolution, and the spectral based are there in the PlanetScope. The implementation of process include the database input and setting, training, and prediction. In addition, it has a certain purpose in the identification and the monitoring of the agricultural and forestry and disease in future. The research results provides a reference for the regional lots monitoring and prevention of disease. Next page. Paper 6, the title Literature Review of the Detection and the Categorization of the Virus Areca Disease Using Image Processing and Machine Learning Approaches. Authors are Punit Beer and Netravati PS. Journal, International Journal of Applied Engineering and Management. This paper research works and contracts on implementing the system using machine learning and image processing to educate farmers on different kinds of diseases on areca nut, detecting the disease in order to reduce the farmer work and increase the revenue of the farmer. Common disease are bud-root and polaraga yellow leaf disease, Aneva roga and foot root, etc. Image pre-processing and image segmentation features exhibition the disease detection. Disease classification are to tasks are done using the image processing and the algorithm. The limitation is the proposed only on areca nut disease is like cholera and dot disease and nut split, etc. According to this soya analysis, the product is good quality and low cost, controls the disease and spread. The proposed method can be implemented with the SVM and random forest of boosting and logic, and results can be compared. Our goal is to educate the farmer by giving the technology to each to find slave in the disease and areca nut and focus on implementing the system using ML and the image processing. The next paper will explain about Kiran.
Thank you, Manjunath. The title of the paper is ID3 algorithm and its improved algorithm in agricultural planting. The author name is Linson and journal is ResearchGate. In this paper, the classification of ID3 algorithm is an important in data mining because it is a greedy algorithm. Each partition only consider local optimization, not global optimization. Pruning is usually required, and data decision tree pruning is overall optimization of this model. The improved algorithm is MIND algorithm. MIND algorithm can reduce the amount of information of unimportant attributes to a certain extent. Compared with classification of ID3 algorithm, it makes the entire operation more independent. which depend on more attributes, thereby improving the classification rules and results, and finding the root node of the decision tree is very easy. This paper improves the problem of attribute value preference in algorithm. Through a large number of experimental verification and comparative experiments, it's concluded that the mind algorithm is more suitable for making decision on agriculture planting, thereby effectively improving the agriculture production and farmer lives. Next paper. The paper 8 title Areca nut yellow leaf disease forecasting using IoT and machine learning. Authors L. Vinod Kannan, M. Sanjeev Kumar. The journal is International Journal of Scientific Research in Engineering and Technologies. In this paper, we have exhibited a system to predict if Areca nut plant is susceptible to disease infection or not by the comparing the data values, which we got by using some IoT sensors like DHT11 and soil humidity, which data values that cause disease in plants that were found by doing historical research. Comparison between these data values are done by using regression algorithm to find similarities in the pattern conditions. A range determined for the scoring of the result and a value greater than 8 on scale 1 to 10 indicates that there is high possibility of disease infection in the region. The output obtained is manually confirmed with experience. This helps to evaluate the model efficiently. The farmers are warned about the possibility of disease so that they can take necessary precautions as a scope for the future work on this model. It can be exhibited to multiple crops and for multiple disease based on the research over various crops and conditions. Next, survey findings. In survey findings, we try to better understanding of the symptoms of YLD and measure taken to minimize. YLD, we come to know the YLD affected regions in Karnataka and the reasons for affecting the disease and spreading of the disease by using the survey finding, and the bacteria that involved in causing yellow leaf disease, for example, phytoplasma like bacteria, and the technique used to detect different diseases of Areca tree using machine learning and image processing. And we come to know the different algorithm which can help us to get some correct output. That's all. Thank you.
Shruti ma'am. Ma'am, any questions?
No questions.
Shashidhar sir?
Sir.
Okay. So from sir side also no questions.